This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1131, recorded on July 12, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody, and Vincent and Rich, and that's everybody. Having a good time. I'm looking out my window today, and it's supposed to be mixed weather. Tomorrow we're supposed to get a lot of rain, but today it's quite pleasant out there, actually. So not too bad considering what's going on everywhere else.
I'm here in New Jersey, not far from you, maybe an hour south of you, Dixon, and
Yes.
it's 28C and 74% humidity. It kind of looks like it wants to rain.
It's going to come down. I think
I
it'll
don't,
come
I don't
down.
really care because I'm inside all the time.
Right.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. A long time no see.
Yeah, it's good to see you again.
Yeah, it's good to be here. Good to be seen. So here in Austin, Texas, believe it or not, it is a mere 85 degrees. It is cloudy. We're sort of hoping for some rain, but, I'm doubtful. It's, less than 50% chance, but that's all okay. It's been, quite a reasonable summer, Austin-wise, so far. All good.
So, Rich, I saw this funny blog, Instagram post the other day. It was a map of Texas, and it said, European trip for cheap Texans. And it had a line going through all the cities in Texas that have European, like Stockholm and Paris.
Odessa.
Gosh, there are like 20 of them. It's amazing.
Yeah, it's pretty amazing.
This is all true.
So funny.
Well, most of the people who settled in Texas were of German extraction, I gather.
There are quite a few, yes.
Yeah.
A hotbed of that stuff is a town just an hour west of us that I really like. It's a good sort of touristy visiting town called Fredericksburg.
Oh, yes.
Where you can go to restaurants and that kind of stuff. But it is also notable as the birthplace of Admiral Nimitz.
That's right.
Okay, and you can visit his house, and they also have a World War II museum of the South Pacific that is absolutely world class. Really great. So good day trip.
Here's some of the towns here. So we got Edinburgh, Stockholm, Fredericksburg, London, Florence, Holland, New Sweden, Dublin, Newcastle, Italy, Texas. Can you believe it? Athens, Naples, Paris, Manchester, Geneva, Belgrade, and Liverpool. Oh my gosh.
There you go.
Don't
live in Liverpool, Texas.
Liverpool, Texas. That's right.
And then there was another post that had a Texas superimposed on Europe. It basically covers all of Europe.
The whole thing.
And it says, this is for people who think they can see Texas, or they can see Texas in a week or something like that. Not that you'd want to see all of Texas, right, Rich?
Well, why not? So I will give Texas a plug here. When we first moved here, one of the things it's been almost 8 years. Within the first year or so, we took a driving trip to Pasadena. And so that drives through West Texas. And West Texas is, in sort of the common mindset, this really boring, barren landscape and that kind of stuff. And so I sort of adopted that as we were going through. On the way back, we stopped at Big Bend National Park.
Oh, that's different.
Okay.
That's another world.
And as is typical of the U.S. national parks, which I really love, they did a fabulous job of really educating you about the landscape and the plants and animals and all that kind of stuff. And I emerged from that with a totally different perspective on the same landscape, okay? Exactly. Recognizing the plants, understanding the habitat and that kind of stuff. So it is what it is, and it's pretty interesting.
When we drove to the Science Mill, that was very nice countryside.
Yeah, that's beautiful countryside. That's driving through the hill country.
I had a girlfriend. I'm sorry, go ahead.
Yeah, go ahead. It's okay.
I had a girlfriend for a long time. She was a Washington-based, sort of a bureaucrat of sorts that worked for the NIH, and we became friends, and then we became intimately associated with each other, let's say. And her mother was living in Texas still. So we used to take, like, every 3 or 4 months, we used to go down there, and we had the most wonderful. There was a wonderful place called Luling. Rich, do you know where Luling
No,
is?
I don't. I think I've
It's
heard of it, but
it's got oil wells that have surrounded the entire town,
yeah.
and some are in the middle of the town also. So she said, now, when we go to eat breakfast, don't be upset when the bacon comes back a little bit oily.
Yeah. Yeah, there
and
are still places in Texas where when you drive through them, you can smell the oil.
and it did, and it did.
Also, Rich
So,
and I drove from Austin to
Galveston.
Galveston. That was
oh,
quite
that's
a
a great
drive. And
drive.
we saw a lot
Yep.
of Texas then as
That's a
well.
good drive. That's a
It
good
was
drive.
good.
And for those who might be listening and interested, the hurricane missed us. It veered east. We were hoping. Ooh, I just heard thunder.
There you
Uh,
go.
We were hoping for some rain. We didn't get it. Some other unfortunate souls got too much.
Right.
So,
Rich, our deep hope is that it was thunder and not something else.
thunder. Trust me.
Thunder. Oh, good. Oh, good.
What else would it be, Dixon?
Oh, something could have blown up.
Yeah.
Oh, okay. Things blow up, Dixon?
Dixon's
Oh, yeah,
in a
that's-
cynical mood today.
No, not
Okay.
necessarily, but
Okay.
you know.
If you like these programs of Microbe TV, including TWiV and many others, science programs, we'd love to have your support to continue producing them. You can go to microbe.tv/contribute. And Microbe TV, our production company, is a 501c3 nonprofit entity, which means your contributions are federal U.S. tax deductible. And today we have the passing of two individuals who Rich is going to tell us about.
Okay, the first I want to talk about is Maxine Singer, a scientist of some repute, died recently at the age of 93. We have an obituary from the Washington Post that we can link in the show notes. She was born in Manhattan, went to Brooklyn High, where she caught her interest in science from a chemistry teacher. How common a theme is that? A teacher determines the course of your life. She also, took up the cause of women in the professional women, okay, and was a force in that throughout her career. She went to Swarthmore, followed by a PhD at Yale, and then assumed a position at the NIH for many years in the area of, it was in the early days of understanding the genetic code. So she made some contributions to the elucidation of the triplet code and later to transposable elements. And she's later in her career moved to the Carnegie Institute and actually branched out into areas of astronomy and et cetera, and was always, as I already said, a force for women in science. She is very notably remembered for her role in the era in the early 70s when recombinant DNA technology was pioneered. And there was a Gordon conference in, 74, 75. It's described in this Post article, where she was a co-chair and raised the issue of that this new technology raised potentially ethical and safety issues that the scientific community needed to take into consideration. And that ultimately precipitated what is commonly known as the Asilomar Conference, where a whole bunch of scientists worldwide gathered to consider the ramifications of recombinant DNA technology and consider what guidelines might be put in place. This resulted in, for a time being, a moratorium on recombinant DNA work, and then while they developed guidelines, and then ultimately self-imposed guidelines that stood for a number of years until it became clear over time that recombinant DNA technology, in fact, was quite a safe activity and actually opened many, many, many doors scientifically. But that stands as a shining light, I think, in scientists' ability to have a sort of an insightful recognition of their impact, potential impact of themselves and their technology on society, and take responsibility for it. So it's quite something. So I note also that survivors include, get this, her husband of 72 years, Daniel Singer, 4 children and 4 graduate children.
By the way,
4 grandchildren, sorry.
This, the Washington Post obit that I'm linked to has a picture of her reading an old style DNA sequencing, Joe.
Yeah.
It's just great.
Yeah, yeah. So thank you, Maxine, for all you did. Another obituary that I think is a first for TWiV and very important to me. Our longtime listener and good friend, Neva Lockett, has died. Those who are longtime listeners to TWiV will recognize the name. Neva was not a scientist. She was a layperson who, from very early on, wrote really wonderful letters to TWiV. She lived in Buda, Texas. When she first, started writing to us, none of us knew where Buda, Texas was. And now I go to Buda, Texas every day to go to the gym.
Is that right?
Neva introduced us to Buda. And through TWiV, really, she became a friend. And my wife and I used to go out to lunch with her occasionally. She came to numerous TWiV functions. She was at TWiV 500. And we recently saw her. She came to the live stream TWiVo in Johnson City just a couple of months ago. So Neva was a special person. In fact, I don't know all that much about Neva other than she was a wonderful person and a great friend. What else do you need to know? So thank you, Neva. Sayonara.
Wow.
Okay. In some news items, first is based on a letter from Robin, who wrote, "As always, enjoying your show. I was wondering if you could address the new ruling from the Supreme Court that rolls back the Chevron doctrine. This was the ruling the government has been working under for decades that stated that agencies had the expertise to interpret and write regulations for laws that Congress passed. I thought of this when Alan stated that it would be up to the agencies to figure out what it means in regards to a ban on gain-of-function research by Congress. This ruling would mean the exact opposite. The lawmakers would need to specifically state each and every regulation that they wish to have enforced, as the vast majority of our lawmakers have little to no science background. And as one party is actively hostile to science, I fear that our regulatory framework will quickly descend into chaos. What are your thoughts on this ruling by an unelected body that also has no scientific expertise? Unelected body, of course, being the Supreme Court with no scientific expertise. So I have a couple of articles that I'll put in the show notes that deals with this. Let's talk about it a little bit. The first one is an article in Science from June 28th. It is written by Jeffrey Mervis. Supreme Court ruling may threaten the role of science in U.S. rulemaking. All right, so this is the Chevron doctrine, which basically it was a 40-year-old doctrine that gave federal agencies considerable leeway in interpreting laws passed by Congress. So the ruling, which was 6-3, means judges should no longer defer to the scientific expertise of those agencies on technical questions and make decisions on their own. And Justice Roberts said, agencies have no special competence in resolving statutory ambiguities. Courts do. And the decision was about herring boats, and Alan Dove talked about this a couple of weeks ago. But this has implications for scientific expertise. So they quote George Washington University law professor Emily Hammond, who says, the idea that generalist judges are now best situated to make these kinds of decisions as compared with scientists, engineers, and technical experts at those agencies really flips on its head decades of expectations about the relative competencies of agencies versus judge. Of course, the conservatives think it's not a problem. And Elena Kagan, right, one of the justices, she had the dissenting opinion. She gave some examples of technical questions that she says the judges cannot answer. For example, the FDA, Food and Drug Administration, must decide what qualifies as a protein when it regulates biological products. And the Fish and Wildlife Service is required to determine what constitutes distinct population segments of imperiled plants or animals to enforce the Endangered Species Act. And she writes, that is what a typical Chevron question looks like, and scientists at the agencies have the knowledge and experience to answer them. It's now a role the court has now claimed for itself as well as for other judges. So there are also some other articles that I found basically from a couple of law firms, and I'll put them in the show notes. One is from Debevoise and Plimpton. It's called Chevron Under Fire. This is in January before the decision came down. What healthcare and life sciences investors should know. And they gave an example of some of the impacts. So there's ongoing litigation challenging approval by the FDA of mifepristone, a drug used for abortions, right? The Fifth Circuit, in a particular decision called Alliance for Hippocratic Medicine versus FDA, held that in 2016 and 2021, the FDA wrongfully loosened the restrictions for patients seeking access to the drug to terminate their pregnancies. In doing so, the Fifth Circuit rejected the agency's scientific judgment that the drug is safe and effective. Okay, so you have to consider that the judges, Supreme Court or otherwise, are now going to decide whether FDA decisions are correct or not. So in this article, they write, such an outcome may inspire other advocacy organizations to seek to overturn FDA approval of other drugs that they oppose. Just go to the courts, and the courts will decide if the FDA decision is right or not. It's not just the FDA; it's many other agencies as well. Anything involving science. So
why have an agency?
Well, agencies have other functions, right?
Well, this is one of the biggest ones. So.
I mean, I think it's crazy from my perspective. I don't know a lot about this. And maybe writing better statutes is part of the solution. But here, what does she say? There's another quote here. I have to find this. This is really good. Basically she says the scientists have the expertise to answer all these things, and now the judges think that they can do it. I can't find where that is. It's in one of these articles, but basically, let's say there's something involving gain of function research. The judges will decide what gain of function is, not scientists. Or maybe there's something involving the origin of SARS-CoV-2. A judge could decide, came from a lab. I read this article in the New York Times. It said it came from a lab, so it came from a lab, and that's it. And whatever ruling is coming up, we're going to take it. So I think this is a disaster for science in the U.S. It's just been getting worse and worse every year, and this makes it even worse. This is the Chevron thing was in act for many, many years, and the conservatives were dying to turn it over, right?
I take a sort of a wait and see attitude towards this. One of the links that you came up with was from this law firm, Arnold and Porter,
right?
Which I thought was quite good, and they don't really take a position on it. They just, as far as I could see, they just sort of describe the situation and some of the potential outcomes without really taking a strong stance. But I came away from that thinking that, you know, this is something that's going to continue to be a contentious issue for a long time. And I also note that in the Supreme Court decision, they do, and this is emphasized in this Arnold and Porter article as well, that it is acknowledged, at least, that some scientific expertise may be of value in making some of these legislative decisions. Okay, so hopefully they would look for some consult on some of this stuff. And it's hard for me to believe that if a situation came up where some really outrageous decision, like, for example, overturning the FDA's scientific evidence on the abortion drug became law, that there wouldn't be at least a wild heap of litigation to follow that. The lawyers are going to have a field day with all of this. Okay. So like I said, I'll wait and see. It's not a happy move, in my opinion, but we'll see what happens. I was thinking of something else, but I forget.
So I found the passage by Kagan that I wanted to talk about. Remember, she said, you're going to have to define what a protein is. And she writes, Chevron has supported regulatory efforts of all kinds, including keeping air and water clean, food and drugs safe, financial markets honest. She argued that abstract analysis by judges can only go so far when defining complex areas of statutory science, like when does a polymer qualify as a protein, right? And she says, I don't know many judges who would feel confident resolving that issue. But the FDA likely has scores of scientists on staff who can think intelligently about it and arrive at a sensible answer. Justice Jackson made a similar point during the oral argument when she asked whether Congress wanted the courts to decide what it means for a study to be adequate or well controlled in the context of a new drug approval. So I understand Rich's position, right? You want to wait and see what happens, but it could be bad.
It could be bad. I don't disagree
Delaying
with
something forever is the same as getting rid of it. And we're seeing a lot of that in these current iterations of all of the transgressions that a certain person has initiated and not taken responsibility for.
Yep.
If you can get the courts to delay and to delay and to delay, it diffuses the issues and makes them non-issues, basically. And then that's how you can take control. You don't have to actually make a decision. You can leave it in the air forever.
Yep. Okay. So there you go, Robin. I hope that helps. And
yeah, I don't know if that, certainly you get some perspective from us. I don't know that it really helps. It's really complicated. To me, this is an example of, science running up against the law. Okay.
Yeah.
The law and science are very, very different disciplines. Okay. And you run up against this stuff all the time where, there's conflicts of interest on both sides, and the resolution is complex. I would presume that the agencies that have been in charge of this stuff in the past are chock-a-block with lawyers who know how to deal with statutes and write the statutes correctly. The issue here is the converse is not necessarily true, that the lawyers are full of scientists, okay, who can contribute to that side. So
But
we'll see how it
but
works
but
out.
judges should not be involved in reversing FDA decisions.
I don't disagree with that.
Now, someone could bring to suit something that the FDA has done. That's fine, but then you need people who are experts testifying, who are
Exactly.
scientists in that, not
Exactly.
just having the judge say this FDA approval is no good. That just doesn't work. Everything
Right.
breaks down.
But
You
you
can
will
imagine
remember, you go ahead.
a world, a U.S. where the judges say, we don't need vaccines anymore.
Yeah. The Scopes trial comes to mind with regards to what is and what is not taught in schools. And we're still up against that.
Good point, Dixon.
Still up against that. Oh, well. And that came to trial, and the judge had to obviously admit, given all of the evidence, that yes, evolution is a process that can be allowed to go forward. But it took heroic efforts on the part of the defendants in order to make that happen. That's a waste of time.
Yep. One more news item. This comes from the New Hampshire Department of Health and Human Services. Case of measles in New Hampshire. Unvaccinated New Hampshire resident. Likely result of exposure to an international traveler with measles who visited Hanover in late June. Follows a confirmed case in Vermont that's also linked to an international traveler. So a 3-person measles outbreak cluster. And they have a list here in this article of where these people were, which is interesting because, peppermint patties on July 1st between 1:30 and 3:30 p.m. So if you were there, maybe you should get vaccinated, right? Isn't that interesting?
You should get vaccinated even if you weren't at peppermint patties.
Yeah, if you've not been vaccinated, you don't have any record, you should.
That is the key word for me in this whole thing, unvaccinated. And of course, my first reaction is it an unvaccinated adult or an unvaccinated child? Because as we have already discussed, there is a window in infancy where it is like the first year, I think, currently in the U.S., when because of maternal immunity, it is not appropriate to vaccinate. So there is a potential window of susceptibility in people. But as I read through this, it seems fairly clear to me that we're talking about an adult. And from my point of view, there's just, unless there's genuinely a medical condition, there's no excuse for being unvaccinated.
Mm-hmm. The state epidemiologist said anybody who is not vaccinated should get vaccinated. 2-dose measles vaccine provides lifelong protection against disease. So the quote from the epidemiologist is interesting. The 2-dose measles vaccine provides lifelong protection in most people, and it's the best protection against measles and complications of infection. Measles is a disease. Now, some people may read that as saying measles virus infection, but it's not. It's the disease that you are protected against. So that's where some misunderstandings can arise, I believe. Okay, we have 2 items for your learning experience today. The first is a comment in Nature Reviews Microbiology, called New WHO Proposed Terminology for Respiratory Pathogen Transmission by Nancy Leung and Donald Milton, who are at the University of Hong Kong. And this is a comment on a WHO report that was issued recently. Let's see, it's reference number 4, Global Technical Consultation Report on Proposed Terminology for Pathogens that Transmit Through the Air. So it's a WHO.
Actually, pardon me.
Yeah.
Donald Milton is at University of Maryland School of Public Health
He's in Maryland.
College Park. Lung is at Hong Kong. Sorry.
Yeah, I got it. That was on the next page. I didn't even go that far. Yeah. Anyway, you can see this WHO document. It's quite long, and Daniel Griffin has talked about it on Clinical Update. And this is a nice summary of it that I thought we could use as a framework to talk about it. And basically, many pathogens are transmitted from person to person. These are respiratory pathogens that are the pathogen goes through the air, let's say, right? And so WHO decided, there was a lot of discussion about transmission of the virus through the air during the pandemic. And so this WHO said this prompted them to convene this technical consultation group to standardize the terminology for transmission modes. Okay. And they say this is important to do to help control transmission in the future, right? By things like masking and airflow and filtration and maybe UVC and all this information. So they say the report has 4 messages. First, it gives you terminology to describe pathogen transmission through the air from one host to another. It should be process-based rather than descriptions of the objects carrying the pathogens. Like, we don't care about the size of the particle that the virus is in. We want to talk about how it goes through the air, right? So that's new terminology, which we'll get to in a minute. Second, the term airborne transmission should be used to mean transmission involving inhalation of pathogens suspended in the air, right? Number 3, inhalation is an important mode over short distances, but it can also be involved in what they call direct deposition, semi-ballistic sprays. We'll get to that in a moment. That's a great word, semi-ballistic sprays. And also contact, right? If you sneeze and your droplets go on a surface and someone touches the surface, that's transmission by contact, but it also involves airborne transmission, right? And then finally, number 4, they have a new term that they define: infectious respiratory particles, or IRPs. And this is a whole range of sizes of droplets. We're not going to use droplet anymore. We're not going to use aerosol anymore. We're just going to say infectious respiratory particles. And they want to shift the focus from the kind of particle, right, a droplet or a droplet nuclei, to the method of transfer, right? So here, the ways that we transmit these respiratory pathogens, we have airborne/inhalation, we have direct deposition, and we have contact. And there's a nice figure that distinguishes between these. It's the one figure in the paper. So, for example, you have 2 people standing near each other, and they're breathing out infectious respiratory particles, IRPs. So that's the new term here. And you can forcefully expel some of these. So if you sneeze and someone's really close to you, your droplets are going to go right to their mucosal surfaces, right? So that's called direct deposition. It really doesn't depend on these IRPs floating in the air for any period of time.
And it does not necessarily involve inhalation.
Right.
It's just landing on a mucosal surface.
That's right.
In your mouth or your nose or your eyes or whatever.
Exactly.
It doesn't necessarily involve inhalation.
Exactly.
These are subtle distinctions,
Very,
but important.
Yes. All right. So that's direct deposition. Then we have airborne transmission inhalation, right? Where the IRPs, the infectious respiratory particles, are exhaled by someone and then they're inhaled by someone else. And then you inhale them and they land somewhere in your respiratory tract and initiate an infection. All right, those are 2 different modes of transmission of these IRPs. But then you have indirect contact, where, let's say you have a cell phone you're handling and you contaminate it. Then you give the phone to someone else and they touch it and pick up a pathogen. That's indirect contact, but it's a fomite basically. Or if you touch someone's hand, that's direct contact, where you transfer it directly. And then there are long-range transmissions, right? So there can be infectious respiratory particles that travel long distances. And measles comes to mind as that. So that's a long range transmission. So far, we've been talking about short range transmission, and then there's touch. So a person could touch a surface and leave, and then someone else can come and touch it again, and that doesn't involve these infectious respiratory particles necessarily.
So one of the things that stood out to me in this is that airborne transmission / inhalation
Hmm.
can apply over both short and long distances.
That's right.
Whereas direct deposition, that is the what was the phrase? Ballistic.
Yeah, ballistic.
These are what we used to call, I think, droplets. Okay. Or usually semi-ballistic trajectory. That's short distances because the IRPs that behave that way
Yeah,
don't are not suspended in the air over long distances.
right.
So inhalation, both short and long, direct deposition, short
Right.
distances.
So as Rich said, airborne transmission inhalation is when these infectious respiratory particles are transferred via inhalation. So, and they this commentary, they say there are two concepts in the report. First is that the inhaled pathogens can land anywhere along the tract. It could be the upper tract, the middle, the lower tract, depends on the pathogen. So transmission is not restricted to the smallest particles that reach the alveoli, for example. They can be other sizes. And secondly, they say there's no typical distance from the source for airborne transmission inhalation. It could be any distance. It could be short or long, as Rich just said. And we used to talk about droplets that fall to the floor, that go 6 feet, that go longer. None of that anymore. It's just any distance, right? And so the direct deposition mode is the ballistic mode, when you forcefully expel IRPs and they go right on the mucosa, as we've already said. But we use different language for that. We call it the direct deposition mode. And also, when surfaces can be contaminated by these infectious respiratory droplets, and so they travel through the air, and that's one part of the process. But there's also direct contact with the surface, right? And the authors say the report claims that this term, infectious respiratory particle, simplifies a highly complex issue, although it states that the term will require training to be understood. And then the authors say it seems to be overly simplified and confusing.
Well, to me, first of all, I mean, it's fairly obvious to me that this all falls out of all the issues that we had during COVID.
Yeah, it does.
Because we used to have discussions all the
Yep.
time about the mode of transmission, the meaning of the word aerosol, the meaning of the word airborne. And this is an attempt to at least standardize some of those definitions, and certainly an attempt to clarify it. And I appreciate that.
Rich, you think they'll leave it up to a judge?
Interesting.
Yeah,
Dixon.
good comment, Dixon.
So I want to go back on the COVID thing and make a couple of other points. One of the points they make in this article is that the concentration of particles is a function not only of the IRPs, probably, but also the distance from the source,
Right.
and how they are expelled from an individual, et cetera. And one of the things that I think of, now check me if I'm wrong on this, but in the early days of COVID, it was fairly clear that it could be transmitted by, what are we calling it now? Direct deposition?
Yes, you mean the ballistic method, right?
Yeah,
Yeah,
ballistic method.
direct deposition, right?
Okay. And that's what precipitated these social distancing measures. Okay. Because the idea was that direct deposition happens only over a short distance, or predominantly over a short distance. And that if you were at a longer distance, you could minimize the chances of transmission through direct deposition. And then there were uncovered situations where it was fairly clear that somebody who was a longer distance away, say in a restaurant or something like that, got infected from somebody in the other side of the restaurant. And also there were physics experiments done suggesting that you could have small
Mm-hmm.
IRPs that could travel some distance and still be infectious.
Right.
And so now we have for COVID what we would call airborne transmission inhalation. I think both still apply, but what is important to me is one thing important to me is what is the frequency of transmission under either of those circumstances? Are you more likely to become infected at short range through direct deposition, and also applicable at short range inhalation? Or are you just as likely to get infected at long range,
Yep.
where you're only talking about airborne transmission inhalation, number one, and number two, the concentration of particles is going to be less. One of the reasons this is relevant is that this becomes, it gets us back into the political realm, is that I've heard discussions in testimony, say with Fauci and that kind of stuff, that says, oh, you said initially that it was just direct deposition, and if we wore masks and social distance, everything would be fine. But you were wrong, okay?
Yes.
All of that stuff was wrong because we discovered that it was airborne.
Right.
Nonsense. Both are true, okay? And I think it's still true, though I would like to see an analysis of this, that social distancing and mask wearing reduces the risk of exposure to that particular pathogen,
Right.
but it doesn't eliminate it because there still can be airborne transmission over a longer
Sure.
distance,
Sure.
though less probable. So let's look at the whole story here. This is not black and white. Okay. I would also like to see a review that goes pathogen by pathogen and talks about the transmission
Yeah, that would be good.
in each case.
I
Okay.
would agree.
Is this direct deposition? Is it indirect contact, direct contact, airborne?
both.
And what are the probabilities of getting infected by any given method, by any pathogen over a distance? Measles is notorious for long range airborne transmission via inhalation. Okay. It's
Right.
really infectious. How about rhinoviruses, Dixon? I mean, Vincent. I remember this Gordon conference that you were probably at. I think we've talked about before, where they did the experiment of having humans. It's like the ferret experiment only
Yeah.
with humans in rooms where they could share the air, but they couldn't contact each other, as opposed
Yep.
to being in the same room and could share fomites and that kind of
That's
stuff.
right.
And the transmission was by either direct contact or fomite,
Yes, that's
not
right.
through the air.
That's
That
absolutely
still
right.
holds, right?
Yeah. Card playing.
Okay.
Have them play cards, right?
Yeah, absolutely. That was, as I recall, a hilarious talk, because it had one photograph of about the direct transmission involves maybe touching your face, touching your nose,
Right.
or whatever,
Right.
and then touching somebody else. And the comment was, think about how often you touch your nose, and then it had a picture that was obviously cooked, but it was great.
Yeah. Right.
Of the audience in a scientific conference, all these guys in white coats, every one of them with their finger in their nose. Okay. What a hilarious talk. It was great. But made me remember it. At any rate,
Yep.
I'd like to see that review. Let's do this pathogen by pathogen.
Yeah, that would be
And
good.
see how it plays out.
So, all right. So now, where do these particles come from? The report says these particles, these infectious respiratory particles, come from fluids that exit an infectious person's mouth and or nose when you breathe, talk, sing, spit, cough, and sneeze. And there are a wide continuous range of particle sizes. So they want to abandon this idea that you have droplets that deposit on exposed mucous membranes, right, in short distance, and droplet nuclei that travel long distances. And when you inhale them, they go way down into your alveoli. I don't want to have that anymore. They said it's very clear that infectious respiratory particles fall to the ground, and as you get farther and farther away from someone who's emitting them, the concentration gets less, unless they're really small particles. But we don't want to classify them into different sizes. And they said all this is consistent with the physics of aerosol generation, right? It's all, what they call aerosol physics, right? All of this makes sense, but they want to simplify it with this term IRP and use this transmission through the air, which combines direct deposition and inhalation. Now, the whole purpose of this report, the WHO says, is to help control outbreaks. But these two individuals make the comment, the report doesn't outline the action items needed to translate the implications of these into policy. And they say we're going to need a decision matrix where we incorporate the mode of transmission, the virulence of the pathogen, host susceptibility, ease of implementation of any measures, communication, accountability, equity to guide interventions. And they say this is too complicated. But we do need to have more discussion about affordable environmental and engineering controls. And we should really be working on this now between pandemics, right? Can we develop new ways to control air and filter it and so forth? That's one thing that they say may come out of this. Now, recently there was a MERS coronavirus outbreak. It was a nosocomial MERS coronavirus outbreak, and it's in a hospital, right? And this report recommended droplet precautions when providing care to symptomatic patients and airborne precautions only when performing aerosol generating procedures. And the authors say this report was made by WHO. They're not even following their own recommendations because they're talking about droplets and aerosols. You're not supposed to do that, right? So that this illustrates a real world complication, right? They say we have patients who are symptomatic, so we should avoid infectious respiratory particle contact with them. So wear masks, for example. But if you're doing a procedure that generates an aerosol, you also have to be careful. And you shouldn't distinguish between droplets and aerosol, right? That's the whole point there. They say basically we have a ways to go before anybody picks up on this. And Daniel is very skeptical. And he said, nobody's ever going to pay attention to any of this. So who knows? Anyway, I thought it would be interesting to talk about these.
I think it's very interesting. I think it at very least recognizes that there's a problem in talking about this and puts at least some definition on it, but we haven't heard the last of it.
No, it's certainly not over. Yeah. Nope. We have a ways to go. Okay. Now we're going to talk about pathogen, a virus, in fact, that seems to involve, infectious respiratory droplets and transmission through the air. And this is, of course, avian influenza virus H5N1. This is a accelerated article preview in Nature, pathogenicity and transmissibility of bovine H5N1 influenza virus. Hot off the press. It's been reviewed and but it hasn't been set into print or anything yet. It's out here for you to see. And so this is coming from the laboratory of Yoshikawa Oka at the University of Wisconsin Madison. But Yoshi also has a laboratory at the University of Tokyo. Shuttles back and forth between the two places.
And I also noticed that in Tokyo, he's Department of Virology at the University of Tokyo.
Right.
The University of Tokyo Pandemic Preparedness, Infection, and Advanced Research Center.
Right.
And the Research Center for Global Viral Diseases.
Yep.
These are research centers that need to be.
And we have 1, 2, 3, 4, 5 first authors: Eisfeld, Biswas, Guan, Gu, Maimura. And besides University of Wisconsin Madison, we have University of Shizuoka, Texas A&M, and of course the University of Tokyo. So we have talked a lot about influenza H5N1 on TWiV lately. Just to review for you what's going on. Remember that we have been finding highly pathogenic avian H5N1 influenza viruses in cows, dairy cattle. First samples detected in March of this year. And that was the first outbreak of H5N1 viruses in cattle. Hadn't been found before. By May, the USDA confirmed 69 infected bovine herds in 9 states. And the spread of the virus is attributed to cattle movement between states. So apparently cattle get moved from one state to another and they bring their viruses with them. And the transmission of this virus among the cattle may occur through contaminated milking equipment and infection through the udder, but the authors point out this has not been confirmed.
The key word there, I read that sentence about 40 times.
May.
That I think could, might, okay?
Right.
There's apparently, as far as I know, no proof for this.
No proof. That's right.
That is an idea.
Right.
And certainly not certainly an idea that has support through the experiments that are done here.
Of course, there have been reports of human infections, right? Dairy workers, farm workers, they have conjunctivitis or respiratory symptoms, generally mild. There have been infections of cats on farms, and these have been fatal. The cats have died, and poultry have also been infected. So the authors point out this is something we need to pay attention to and look at the risks, what's going on here.
So highly pathogenic H5N1 flu has been on the radar for almost decades, right?
Oh, yes.
And there are occasional spillovers into humans that there's no human-to-human transmission that's well documented. Some of these are fatal cases. I think there's a lot more exposure if you do serosurveys of the human population. So this is happening all the time. But this is unusual. This virus has apparently some different characteristics that they're exploring here. The concern has always been, okay, is this the next pandemic? And so this sort of study is really important in looking at that, because this has some characteristics that could make you at least raise your eyebrows relative to pandemic potential.
So, Rich, are you insinuating that this virus might have mutated?
Ooh. Dixon. Very good. All right. So these viruses circulating in cattle in the U.S. currently are related to H5N1 viruses circulating in North American wild birds. They belong to a specific HA clade called 2.3.4.4b, and they were introduced into North America in late 2021 through the transatlantic flyway from Europe. Yeah, birds fly across the Atlantic from Europe to North America. And so we didn't have these here before. And those viruses, once they arrived here, they reassorted with other avian influenza viruses, and they say there are now multiple genotypes that have spread throughout the American continent. There have been outbreaks in wild birds, there have been outbreaks in sea mammals, and some of them high mortality rates and suspected transmission among sea mammals.
So this was an ambiguous sentence to me that high mortality rates, is that certainly that applies to the wild birds. Does it apply to the sea mammals as well?
Yeah.
Are they dying?
Yeah, there was an outbreak that I'm thinking of sea lions in South America. We talked about it on a TWiV that we did in Baltimore a couple of years ago, and there was high mortality there.
Okay.
And in fact, Ron Fouché said that's what worries him about these viruses, that they can cause high lethality in wild non-human mammals. Yeah.
Okay.
Sea mammals. So, typically, as Rich said, these H5N1 viruses, which we've known since the late 90s, they've infected, maybe 1,000 or so, or caused disease in 1,000 or so people. But they don't seem to transmit among people. There's very little evidence that they transmit among mammals. And so the key here with these bovine viruses is are they transmitting among mammals? And can we study that in the laboratory? So that's what they did in this paper. They took a bovine H5N1 virus that they got from an infected dairy cow in New Mexico in the US, and they studied it in the lab in mice and in ferrets. They looked at its transmission and they looked at its receptor specificity, a nice set of basic virology experiments. There's no transcriptomics, there's no dot plots, there's no modeling. It's straight, cool virology.
Yeah, this is actually reads very well. This is a nicely written paper and was, really easy to assimilate.
Yep. All right, so remember, we have this isolate of H5N1 from a cow in New Mexico, US. So they previously have shown that if you take milk from an H5N1 infected cow and feed it to mice, the mice can get infected. Actually, we mentioned that on TWiV a while ago. So they repeat this experiment with different volumes of milk from 25, 10, 5, and 1 microliters of volume, which corresponds to from 3 times 10 to the 3 PFU, right? It's about 3,000 PFU, all the way down to like 100 PFU. So they infect the mice, they feed it to them, and they look at body weight loss and survivability over 14 days, and they determine virus titers in the lungs, the nasal turbinates, and the brain. So the brain is it's a proxy for saying, can the virus spread away from the respiratory tract? So the mice that got the highest volume, 10 or 25 microliters, they had substantial weight loss. Some of them died. There's high virus titers in the nasal turbinate, the lung, and the brain tissue, so it's able to spread systemically. And they have milk from uninfected cows as a control, and these don't do anything to the mice, right? The mice say, yeah, that's delicious. Give me more. Oral inoculation. And 1 microliter of milk, which had 130 PFU, didn't do anything to the mice. No disease, no virus replication. And all the survivors did not make antibodies to the virus. It's interesting.
Yeah, that caught my attention.
I don't know why that
it.
is. I'm not sure.
That doesn't make any sense to me.
You think that, if you're shedding virus, it's replicating and you should be making an immune response. And they're looking at how many days, 14 days should be enough to make antibodies, right, Dixon?
Right. Now, let me ask you this: that they initially discovered that they were infected, at least.
Yeah.
They had to know that.
Yes, they were infected.
Yeah.
So, hooray for T cells.
Yeah, that's what I was going to say. What are the correlates of protection for flu?
Well, severe disease, probably T cells. Yeah. Yep.
And we're going to find out later on in ferrets, they do mount an immune response.
Right.
Or an antibody response.
A week.
So
immune
Okay.
response.
So that was after feeding mice virus. So you feed mice virus, it can reproduce in the nasal cavity, the brain, and the lungs. How about intranasal infection, right? That's how you would typically inhale influenza viruses. So they give mice intranasal virus, and they give them different doses, right? Nothing to up to 1 million PFU. And any mouse that got more than 1,000 PFU, they died. Some of them that got 10 to the 2 or 10 to the 1 survived. And the mouse lethal dose, 50%. So the amount of virus that'll kill 50% of mice, they calculated for this virus, 31.6 PFU.
So that's pretty nasty by my reckoning.
It is, yeah. It is pretty nasty. It's
at least in mice.
Doesn't take a lot of virus to.
Now this was grown up purified virus, right? Rather than this, we're not talking milk anymore.
No, there's no more milk here.
Okay. So we show that we can transfer it by milk, but we also now do a more refined experiment with purified virus and get
Right.
an LD50,
Right.
basically.
Exactly. All right. So this is comparable to 2 other clade H5N1 viruses that were isolated during a mink outbreak in Spain. Okay. So now where does the virus reproduce after intranasal infection? They inoculate mice. This time they put 10 to the 3 so the mice survive. Of cow H5N1, they have another isolate of H5N1. Then they have a pandemic H1N1 for comparison. It'll be a human virus. They inoculate the mice, and then they take different tissues and determine the virus titer. And so both the viruses cause systemic infections. They get respiratory replication and non-respiratory replication, including mammary glands, teats, muscle tissues of the leg and the back. It was found in the eye of a single mouse. So that's both H5N1 viruses do that. But the H1N1 don't spread systemically in these animals. They detect them only in the respiratory tissue. So the H5N1s are clearly pantropic. They can replicate in more tissues than the respiratory tract. Both of the H5N1s can be found in the eyes, mammary glands, muscle, and that's consistent with other reports. So cows get infected in the mammary glands, and the humans are getting conjunctivitis and respiratory symptoms as well.
So this, they're comparing the cow virus to something called VN12. Let me see.
Yeah, VN is another H5 isolate.
It's another H5N1, but it's not from a cow.
It's from Vietnam. It's probably a human.
It's from a human. They say
Yeah.
a typical avian H5N1 virus isolated from a human.
Right.
Okay. VN1203.
Right.
There we go.
Okie dokie. Let's see. So they say they also find the 2 H5N1s in the blood, but not the H1N1. And they say maybe the spread to the non-respiratory tissues occurs through viremia, virus in the blood. Right. Then they move to ferrets, another model for influenza viruses. They intranasally infect ferrets with the 2 H5N1s, and they look at where the virus is reproduced. The ferrets that are infected have elevated body temperatures. They have weight loss, so they show clinical symptoms, right? The virus replicates in both the upper and the lower respiratory tract and spreads to non-respiratory organs, eyes, brain, colon, liver, spleen, kidney, heart in some of the ferrets. Also found in mammary glands and teats, but not in every animal. No virus in blood or muscle tissue. And they don't know why that is in the ferret. Maybe things are different in the ferret from in the mice. And so they say the results support the idea that mammary gland and or teat tropism are features of a mammalian infection with H5N1, and they are not a specific characteristic of the virus coming from cows.
Yeah, I think that's really important.
Yeah.
That's a major message of this. With respect to these assays, they do not discover a distinction between the cow virus and another human H5N1 virus.
Right.
Though, as we said in the introduction, there are observations that suggest that the cow virus, at least in some respects, is different than other H5N1
Right.
viruses that we've seen. But these particular experiments don't reveal exactly what that difference might be.
Right. So the virus from cows can induce, or after oral ingestion or respiratory infection, can induce severe disease in mice or ferrets, and infection by the oral or respiratory route can lead to systemic spread of the virus to non-respiratory tissues. All right. So the next set of experiments are about transmission. The first, as you know, the virus has been found in milk of dairy cattle. And if they feed this to mice, the mice can get infected, as we've shown in this paper. So they ask if lactating mice can transfer the virus to suckling offspring or adult contact animals, right? So they intranasally infect lactating females with cow H5N1 and then put the pups back in the cage, or they put them in another cage with non-lactating female adults. And then after certain days, 4, 7, and 9 days after that, they look in different organs to see where the virus is. Right. So at day 4 post-infection, the 5 out of the 6 lactating females had virus replication in the respiratory tissues. That's good because you had inoculated them. No virus in the brain, no virus in the mammary glands at 4 days. None of the 25 pups that they put with these lactating females had any virus in the brain, lung, intestines, and none of the 3 adult contacts had any detectable virus in the nasal turbinate, lung, or brain. Right. So at 4 days, there's no transmission from the lactating female to either pups or other adults. At day 7, there's more virus in the lung and nasal turbinate of these lactating females. And now they can see transmission to pups. So 4 pups of the 24 are become infected, but none of the adult contacts got infected. So again, there's probably not enough virus there to infect them. In day 9, after infection of the lactating females, all of them, you can find virus in the respiratory tissues and in the brain, mammary glands. 11 of 30 pups became infected, and in them, you can also find virus in mammary gland of the mothers. But the adults still on day 9 did not get transmitted. The virus didn't transmit to the adults. So they conclude that the virus H5N1 can be transmitted from lactating females to pups, but not to adults with whom they have direct contact. And they say maybe the pups got it through the milk. It's hard for them to distinguish milk versus other ways of transmission.
Yeah, you can't. This is a really tough experiment to interpret.
Right.
Because, it could be transferred through the milk, but the pups have all sorts of other contact with the infected moms as well. So it's consistent with transmission through the milk, but it's not a smoking gun.
That's right. Next experiment, transmission in ferrets. So as of this writing, we don't know if these viruses transmit well among mammals, these cattle H5N1. So they do a droplet transmission experiment in ferrets. So they infect ferrets intranasally with the H5N1 virus or the H1N1, which is known to transmit among ferrets. And then you put them in cages next to uninfected animals. So they can't touch each other, but air can go between cages about 5 centimeters apart. And so the infected ferrets, you find high virus titers in nasal swabs over multiple days with the H5 or the H1 viruses. Only the exposed animals in the H1N group had signs of clinical disease and virus in the nasal swab. The cow H5N1 virus does not transmit efficiently via this mode in ferrets. They did antibody assays from the ferrets that survived, and they find high neutralization titers for all the infected ferrets and the exposed animals in the H1N1 group. No virus was detected in any of the animals exposed to the H5N1 infected ferrets, although one of the 4 animals who were exposed had a positive but low antibody titer to the HA. So they've concluded that they didn't see any disease in the contact ferrets with the H5N1, but there's some evidence that they got infected. So they say it's probably really inefficient transmission in ferrets, right, of the H5N1 virus, which may explain so far why it hasn't done much in people, right? All right, the last experiment is really interesting, in my opinion. You want to know what's the receptor for this H5N1 virus? So human influenza A viruses bind sialic acids, a sugar with a linkage to the next sugar called alpha-2,6. It's a specific chemical linkage. And avian influenza viruses prefer alpha-2,3 linked sialic acids. So they have an assay that can say what kind of sialic acid linkage does the virus prefer. They measure virus binding to alpha-2,3 or alpha-2,6 linked sialic acid. The human H1N1 virus clearly prefers alpha-2,6. The Vietnam H5N1 clearly prefers alpha-2,3. The cow likes them both. The cow H5N1 virus will bind either alpha-2,3 or alpha-2,6. So that means our upper tracts have alpha-2,6 linked sialic acids, but not alpha-2,3 except for the eye. So that means that humans could be infected in the upper tract with these viruses. And the Vietnam H5N1 only only alpha-2,3 binding, sialic acid binding. So they say these cattle viruses, these cattle H5N1 somehow are different because they can bind both alpha-2,3 and alpha-2,6. So I was surprised about that.
Yeah.
The mouse the cow mammary gland has both. And I'm surprised because, well, it hasn't transmitted among humans. So obviously binding to alpha-2,6 is not enough for transmission among humans, right? You need something else. So those are the experiments. I think they're very interesting. Inefficient transmission among ferrets, but not in mice. And they do transmit well among mice, and the ability to bind both alpha-2,3 and alpha-2,6. Now, as far as transmission among mammals goes, so there's obviously some cow-to-cow transmission going on, right? Because they think it's going from herd to herd, but they haven't proved it yet, as we said from the sentence in the introduction. There was an outbreak of H5N1 in mink in Spain and sea mammals in South America. There's some evidence that there may have been animal-to-animal transmission, but it's really hard to prove, right? It's just an observational exposure. The CDC has reported respiratory droplet transmission in ferrets of an H5N1 from one of these cattle farm workers in Texas. So that corroborates the evidence here. They say it was limited respiratory droplet transmission. And so they say the discovery that these H5s may transmit among mammals is a paradigm shift and increases the pandemic potential of these viruses. So what else do you need besides binding to alpha-2,6 sialic acids? Well, there's another change in a viral protein, PB2E627K, right? Glutamic acid to lysine at amino acid 627 of one of the viral polymerase proteins. That allows efficient replication of avian influenza viruses in mammals, and that has not emerged in that is not present in this isolate that they studied. However, they say that change was found in a Texas infected farm worker. So the virus may be changing at that position, which would make it more likely to be able to reproduce well in mammals and maybe transmit.
Am I correct that polymerase mutation is one of the ones that turned up in the gain of function experiments?
No, it is. I think it may have. I think they introduced it before they did the passaging.
Okay.
But do you remember we had the TWiV at ASV a couple of years ago? You were there. Science can break your heart. Remember that?
Yeah.
What's his
I don't
name?
remember the details.
I'm looking up his name.
Yeah.
TWiV. He was one of the people who discovered that. Andy Meal.
Andy Meal. Okay.
He works on that.
That was a really good TWiV.
It was very good. Andy Meal and Pamela Bjorkman were involved in that.
I'd like to express a worry of this
Yeah.
virus in cattle.
Yep.
Based on a recent television documentary that I saw, where the first peoples of North America are busy reintroducing buffalo
Uh-huh.
to their properties and allowing them to roam free.
Huh.
They're not fenced in at all. They want things to return to the way they were, before the railroads were put through, I guess. I don't know. I don't think so, but they certainly want a lot more buffalo than there is now. And I'm just wondering what the bovine avian influenza will do to the buffalo, because that's such a naive population of mammals that have never encountered this before.
Yeah,
They're just
good point.
recently exploded. There's about 400,000 of them now, which is a lot of buffalo, but not as many as 3 or 30 million that there used to be. What's going to happen here? I don't know.
Interesting.
Yeah.
And to me, that raises the question of what really is the mode of transmission
Here.
in the cattle.
Yep.
Because you're not going to catch me trying to milk a buffalo anytime soon.
No.
Okay.
No. And they don't interbreed. They do not interbreed, although you can, I think, artificially interbreed them. They don't naturally interbreed, I should say. So
running through my mind through all of this is, we talk about pandemic preparedness.
Mm-hmm.
How do you identify what the potential pandemic viruses are? And how do you characterize their pandemic potential? And these experiments are exactly
Yes.
the kind of thing that you do. And I would say that in the past, when we've talked about H5N1, I've in some ways kind of blown it off because of the lack of evidence for any human-to-human transmission or anything like that. But this is different. There seems to me to be an evolution going on.
Here.
So the authors conclude, they say, currently we don't know whether this dual receptor binding specificity reflects adaptive changes in cattle or is also a trait of other North American H5N1 viruses, right? That has to be looked at. And our study shows that bovine H5N1 may differ from previously circulating H5N1s by possessing dual human-avian receptor binding specificity with limited respiratory droplet transmission in ferrets. So changes have been happening in these H5N1 viruses. And as they say, we need to keep an eye on this because we don't know where it's heading. Okay.
Okay.
Do a couple of emails.
First one.
Go take that first one, Rich.
Okay. Martin writes, Dear Twivers, happy birthday to Rich Condit today. That must have been on June 29th.
That's right.
Looking forward to hearing him again soon on TWiV. All the best to Jarl Martin in Ilsworth, UK. Well, Martin, thank you, number one. Number two, how the heck did you know it was my birthday? Number three, on my birthday, I was a very short distance from you. I was in Soho in London. And I have to tell you that my birthday in Soho coincided with the climax day of Pride week or month or I'm not sure what in London, where they had
Oh,
events
wow.
in Trafalgar Square with a stage and musicians and stuff like that. And I don't know how much you know about London and its various areas, but Pride Day in Soho was really something. I've never been to Carnival, okay, or Mardi Gras, but this has got to be something like it. I'll tell you, it was shoulder to shoulder partiers of the wildest assortment of individuals and costumes that I have ever seen. And it was wonderful, absolutely wonderful. People were having just a great time. It's not my idea of a party,
Mhm.
but there you go. I'm an old sedate guy now, but I loved it. Okay, it was great. It was great to experience. And he says all the best to Jarl. Why apostrophe ARL? That's got to be some sort of British perversion of y'all. Either that or a misprint. At any rate, thank you, Martin.
It could be
I had a great
a
day.
misprint. Yeah.
Could be a miss.
Dixon.
Yes. Hunter writes, TWiV team, keep up the great work that we have all grown to value so much. The coverage of bovine influenza H5N1 has been great. More cows is always a good thing. Another example of politics influencing science perceptions, and then he gives a reference. As a bovine veterinarian for 41 years, it astounds me that people do not appreciate the value of pasteurization. We strongly urged all of our dairy clients to feed on farm pasteurized milk to their calves. One suggestion for Dr. Spindler and ASV, make a visit to a dairy farm near the next meeting site. Seeing how the modern dairy cow is cared for and handled might change some hearts and minds about agriculture today. Hunter Lang, DVM, retired bovine practitioner.
So this
Cool.
article is a survey that says under half of Americans know that raw milk is less safe to drink than pasteurized milk.
Right.
They did a survey.
Right.
How effective is
Incredible.
pasteurization at killing bacteria and viruses? 54% say it's very effective. 22 say it's somewhat effective. 20% not sure.
There's bacteria in milk?
And then the other question is raw milk safer, less safe, or just as safe to drink? 47% of people think it's less safe to drink, and 30% are not sure, and 15% say it's just as safe as pasteurized. Oh my gosh.
Mm, 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 mm.
It's kind of scary,
And
right?
why do you think they call it pasteurized?
Yeah, Louis Pasteur.
Louis Pasteur, exactly.
Charmaine writes, tell Rich, I think his photo on the TWiV website is great. It's a photo we took in Austin, right?
Yeah.
Not too long ago. Yeah. So he's got his ripping his shirt open, showing this spike structure on his t-shirt. Very cool. Also, you need to get a pic of Brienne up there. Yeah, I know. We asked Brienne, but she hasn't sent us one. Thank you so much for that list of episodes on the origin of SARS-CoV-2. I've been wondering for a while how to do a search for that subject or any subject, and now have something to forward to folks who give me pushback on that issue. Also, I would have added Laurie Garrett's episode. Granted, she's not a scientist, but her not too in the weeds approach and her tell it like it is personality would appeal to many non scientists and be more accessible to some. As I recall, she talked about finding the RNA in the drains, practices, and layout at the market, and the wildlife trade with Southeast Asia, which helped fill in the picture. Which episode was that? Let's look. Laurie Garrett, TWiV. That would be 773. Laurie Garrett, Pandemic Prophet. I think that's the only one she's been on. Now, there's another one called Public Health with Laurie Garrett, Wastewater Testing. It wasn't an episode of TWiV. Yeah, it was released on October 4th, 2022. And then 773 was June 2021. That was her first appearance. I agree with you about her communication skills.
She also came to the party at the
She
incubator.
did. She did.
And now that we're searching for names associated with TWiV, I remember if you Google search Neva TWiV,
Yeah.
you can find some of her letters. Okay. Going back, to TWiV 190.
Mm. Yep. She's got a lot of letters in there.
Yeah.
Let's see here. Well, we only have 2 left, so let's do those, or 3. It's Dixon. You're next. You're muted, I think.
You're
Let
muted.
me unmute. Robert writes, starts off with a reference. The concept was taught to PNS students in the late 1960s. I taught there from 71 to 1997. Is it still a valid theory? Might annual flu shots achieve the same, or do they provide little long-term memories for the immune system? Take it, Vincent.
Oh, Robert is a PNS graduate of 1972. Cool.
Yes, he is. That's the second year I was there.
Look at that.
He was there.
So this is an article I talked about the other day. Apparently, there's this thing with influenza infections, originally antigenic sin, right? Or immune imprinting, which is a non-religious version. Your first encounter with influenza is really important because all your subsequent ones, you make a memory response to that first one. And so it turns out this study that is reviewed in this article, that certain people over an age have antibodies that protect them against severe H5N1 disease due to being infected with a virus early on that was somehow related to H5N1, mostly older people. Now, Robert asks, do the annual flu shots achieve this? No, they don't, because unless you're over 65, they could cause a memory response if you had been infected earlier with a H5 related virus, right? But not if you're younger than that. That's according to this study.
As I recall, this phenomenon played a role in the 2009 pandemic. Is that correct?
That's correct. That's absolutely correct.
Because there was some relationship between that virus and the 1918 influenza. Yes. Okay, and some older patients, people who had been exposed to that, had greater immunity to the 2009
That's absolutely
pandemic.
right.
Very interesting. Now, what did you call? I can only remember
immune imprinting.
Immune imprinting. Is that a flu-specific thing?
No. It was discovered with flu, but it's not really flu-specific.
Right. I wouldn't think so. Okay.
Who's next? Rich.
Charmaine writes, if you do have an episode on science and politics, I recommend Richard Pan. He is a pediatrician who was in the California State Senate and authored bills, among others, to remove the religious and philosophical exemptions from required school vaccinations and also make it illegal for anyone to harass someone on their way to receiving a vaccine. Here is his Wikipedia page, and she gives a link to Richard Pan's Wikipedia page. This is familiar. I remember this guy coming up in the news with respect to some of this legislation. Cool.
Yeah, we made a call a couple of weeks ago about getting people who could talk about the intersection of science and politics. So people are sending in their suggestions, and I'm reading the letters. That way it gets into the record here. And then this last one is from Greg, has a suggestion. My suggestion for a guest to discuss politics and science is Naomi Oreskes, historian of science at Harvard. Her website describes her area of research as Anyotology, the political economy of scientific knowledge. She is focused on earth and environmental sciences, if virology and public health, science is too far from her expertise. I suspect she could recommend someone with relevant expertise. I don't know, but I suspect that she and other historians will say that the golden ages of science, where politics did not impact science, have been rare if they have existed at all. Scientists are human and are influenced by the ideas and prejudices circulating in their communities and depend on funding and to some extent live in a world governed by the golden rule. The man with the gold makes the rules. One role of scientific societies such as AAAS is to advocate on behalf of science, and that is a political act. In a somewhat related vein, I'd be interested in TWiV's take on a recent article by and podcast with Dr. Vinay Prasad, MD at the University of San Francisco, who claims that scientists involved in the approval of the Pfizer COVID-19 vaccine delayed the reporting results so that the EUA could not be approved until after the 2020 election so that it would not benefit Trump. He also critically discusses vaccine mandates and mask mandates, vaccine harms, and what he considers misleading communication from Fauci and others. I think Prasad was misleading about Fauci's comments, and I tried to make that case in the comment section of the EconoTalk podcast. If he's invited on TWiV, I suspect the discussion would be contentious and it might be interesting and informative. Obviously, the TWiV team would need to review his writing and comments before inviting him. I'm interested in what the TWiV team thinks about Prasad's comments. All right, so let me pause here and say that Paul Offit talked about this incident. So during the trials of the Pfizer and Moderna COVID vaccines, late in the process, Trump called the head of the FDA into the White House. And Paul Offit tells this story. And he said he was screaming at him to release the vaccines before the election. And the head of the FDA said, we don't have the safety studies all done yet. We need to get those done, and then we will release it. And he was screamed at, and yet he held his ground. So that's the version of the story. They didn't purposefully delay the results. They waited until the safety studies were appropriately done. And I think that was the right thing to do. And I think it was the same FDA head who let Trump push him into approving hydroxychloroquine, which was a huge mistake and had to be reversed. And maybe he had learned from that. Anyway. If he's invited on TWiV, I suspect the discussion would be contentious, might be interesting and informative. Obviously, the TWiV team would need to review his writing and comments prior. I already read that. Sorry. Dr. Jay Bhattacharya, Stanford U, seems to share some perspectives with Prasad, so I've added a link to an interview with him. One of the challenges of discussing science versus politics is that science rarely speaks with one voice. There's politics inside science, but the aim should be to sort out differences empirically. There are often different ways of collecting, analyzing, and interpreting data, and coming to agreement sometimes takes decades. And indeed, thanks for your good work and best regards. All right. Time for some picks of the week. Dixon, what do you have for us?
Right. Well, I'm thrilled to report that the city of Stockholm has a plan which has been put into action already to construct a portion of their city which will contain 7,000 office spaces, 2,000 homes, and cover 250,000 square meters. The whole area will be made out of mass timber or cross-laminated timber, as I refer to it in my book. The district will feature nature-informed elements and be designed to have the feel of a forest. Now, I wish I had this data with me when I was writing my book, and I wish they had my book when they were planning this episode of extreme environmentalism, I would think. Space in any city is limited, and here they are devoting 250,000 square meters to an idea whose time has come. And also, you must know that Sweden has a very large lumber industry, and they're looking for a way to involve them in the future of urban life. And no better way that I can think of course, is to start constructing with cross-laminated timber. And that's what this article is all about.
This is very cool. I like this.
Yeah, thank you.
The pictures are wonderful.
They're very
What it does
seductive.
to me is, I am too close to the news, and I can spend a lot of time sort of depressed
Yeah,
over
yeah,
the directions that we're
you're right.
taking, and climate change and the future of the planet and all that kind of stuff. But in
Right,
the back of my mind, I try to remind myself humans are very clever,
indeed.
and uh, this is a good example of that. So
I would
it's
agree.
not we are not necessarily going to collapse. I think there's
No,
going to be crises out there that will hopefully help people come to their senses.
right.
But this is sensible.
It is.
Very cool.
I'm fond of saying that human beings are geniuses at getting themselves out of messes that they created.
Yeah.
Thanks. I was in Stockholm not too long ago. I didn't know about this,
Yeah,
but
it's too bad.
it hasn't started to be constructed yet.
No, this is a
It's
Plan,
plan.
yeah,
it's in the works and it will happen. And there's no doubt about the fact.
yeah,
Otherwise, I wouldn't have posted it because their lumber industry will benefit
yeah.
tremendously
It's
from
cool.
this.
Rich, what do you have for us?
I have a book called The Wide, Wide Sea by Hampton Sides. And this is nonfiction, and it's a description of Captain James Cook's third global voyage. The goal of which was, as for many before him, to discover the Northwest Passage. And he wound up convincing himself, at least, that it did not exist by sailing all the way up between Alaska, through the Bering Strait between Alaska and Russia, to the polar ice cap, and sailing all along its boundary from Alaska to Russia and back. Okay. And said, man, sorry, dudes, but there's no way through.
Hmm.
Also notable on this trip was that he, in quotes, discovered, relative to the Western world, Hawaii. He was the first recorded Westerner to land in Hawaii on his way out on this trip, landed in Kauai. And he decided, after probing the ice cap, said, it's kind of late during the summer, and I'd like to come back at the peak of summer. So this winter, I need to go somewhere where we can refresh ourselves and then revisit. So he went back to Hawaii. This time, landed on the Big Island, and there was an unfortunate incident that happened there between he and his crew and the natives that resulted in his being killed, along with, I should say, a lot of the natives. There was a conflict that happened that resulted in his death, and his successor on the cruise actually went back to the ice cap and then went home. At any rate, this is a very well written description of that whole voyage and a worthy read.
Nice. That's pretty cool.
I've now read, I picked this earlier, the his actual log of his first voyage, the goal of which was to record the transit of Venus in Tahiti. And I've not read his second voyage, the goal of which was to determine whether or not there was a land mass at the South Pole. And then this is his third voyage.
Cool.
Quite an adventurer.
Okay, my pick. So I don't usually watch sports. I never watch sports, but I think the Tour de France is amazing. It's a bicycle race, of course, in France, and it's ongoing now. And I found a YouTube channel where they give summaries because, they're 4 hours sometimes. You don't want to watch the whole thing. And this link is stage 12 of NBC's coverage, which I think is pretty interesting. But I think it's an amazing race of endurance and strategy, just to see how the riders, ride behind each other, and then at the last minute they pull out. And the teams, support each other. They draft in front of the leader, and I just think that's very interesting and makes it more than a sport. So Tour de France. I'm linking to stage 12, but You can find any of the stages there. It's really cool. It goes through France and it goes through flat areas. It goes through incredibly mountainous areas. There was a stage the other day where they ran on gravel. Oh my God, they're riding bicycles on gravel, and all this dust is
Right.
being kicked up, and there are all these cars riding, and there are accidents where people get hurt and so forth. Really interesting. I like it.
Crazy.
We have a listener pick from Amy. Regards from an appreciative TWiV listener. I'm sending a 3-minute animation of the work of my friend Merav Ofer, professor of astrophysics at Boston University. The animation is based on a paper she recently published in Nature Astronomy. She gives a YouTube link for that. In it, she discusses how a collision between our solar system and an interstellar cloud of cold gas may have cooled the Earth's climate, potentially impacting the history of life on Earth and viruses. Thank you for being a beacon of reason and light during the COVID pandemic. Best regards, Amy. Thank you for that. That's pretty cool. All right, that'll do it for TWiV 1131 1131. You can find the show notes at microbe.tv/TWiV. Send your questions, comments, and pics to twiv@microbe.tv. If you like our work, please consider supporting us financially. Go to microbe.tv/contribute. Dixon Despommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Thank you, Vincent and Rich. It was wonderful sharing another trio
Last
of
week we had
Twivers.
a duo. We had me and Alan.
Yeah,
Oh
wow.
my,
Can't
oh
get any
my,
less
oh
than
my,
that.
oh
We
my.
don't do ones.
No, well, you could. You really could
No,
do
it's
one
supposed
all
to
by
be a
yourself, but
conversation, you know.
I understand that. Well,
Can have a conversation with myself. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a
I'm
good
Vincent
time.
Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.